come together and have three distinguished panelists in our webinar. We're going to explore how gut microbiome and aging are interconnected and how generally microbiome is affecting our health and the process of aging. Today we have three speakers with us. Firstly, we have Professor Sandhya Visweswariah. She's a former chair and honorary professor at the Indian Institute of Science, an adjunct professor at the University of Bergen in Norway, honorary visiting professor at Imperial College London, recipient of various national and international awards, and she's also a National Science Chair and J.C. Bose Fellow. And her work and her research interest focuses on gut biology, on signaling in bacteria and in the human gut, and how bacterial toxins cause disease and diarrhea, and in general, how the microbiome interacts with the gut. So it's very nice to have you here, Sandhya. We're looking forward to your inputs, and I hope the students and the audience can learn a lot and get to know about your work and your views on this. We also have Dr. Vishal Rao. Dr. Vishal, nice to see you again, and welcome to the webinar.
Thank you.
Dr. Vishal is a head and neck surgeon at 𝗥𝗲𝗴𝗂𝗏 in Bangalore. He has vast experience in oncology. He's a surgeon. He's been a pioneer in voice prosthesis. He's literally been giving voice to cancer patients as an inventor of the Om voice prosthesis device. Dr. Vishal is also a recipient of multiple international and national awards. He's been a visiting scholar at the University of Pittsburgh School of Medicine. He's also been a director at the Cancer Prevention and Tobacco Control Project with WHO, and he's been a fellow at the Tata Memorial Hospital, Mumbai. So vast experience, enormous, a lot to learn and know from Dr. Vishal on this topic. So welcome again, and
thank you.
Looking forward to your inputs. We also have Mr. Darshit Patel with us. Darshit is the founder at 𝗥𝗲𝗴𝗂𝗏. He's also the chief scientific officer there. He completed his master's in drug design and biomedical science from the Edinburgh Napier University. He's been a visiting lecturer at Bharati Vidyapith in Pune. And 𝗥𝗲𝗴𝗂𝗏 has been at the forefront of gut microbiome research in India. Darshit has pioneered many aspects of longevity research and healthcare testing and biomarker testing. He's also a keen observer of technology in the healthcare and aging industry. So today he's going to talk to us about his endeavors in understanding the process of aging and generally applying cutting-edge research in a more fundamental as well as an applied way in the context of India. So great to have all of you here. And I guess the audience is still joining, but it's really nice to see so many people also join us. It's really encouraging. We're going to start briefly with a brief round of introduction from our three panelists. First Sandhya, then Dr. Vishal, and then Darshit. And I request them to briefly talk about what their interests are, how they are connected with microbiome, and what they work on. And then we can start our discussion and questions. Sandhya?
Thank you, Shantanu. So the focus of research in my laboratory has been related to a particular receptor, which is expressed in the gut and is the target for bacterial enterotoxins that cause watery diarrhea that almost every one of us must have experienced at some time or other in our lives. This receptor brings about its action by bringing changes in the epithelial cell of the gut, resulting in iron and fluid secretion, which is basically the diarrhea that we all experience. Now, what became interesting around a decade ago is that mutations in this receptor were correlated with inflammatory bowel disease and Crohn's disease. And we were the first to describe these mutations, characterize these variants found in the human genome. And as a result of our research, this gene has now been part of the panel of genes that are looked at amongst the many hundred that are important for Crohn's disease and inflammatory bowel disease. Now, in order to be able to understand why this receptor causes this kind of inflammation, we've developed various animal models which express different forms of the receptor. And we've been now working with mouse models to be able to understand how they might bring about the inflammatory changes that happen in the gut. Now, you can't talk of the gut without thinking about the gut microbiome. And what we do find in these mouse models, and indeed in the patients that harbor mutations in this receptor, are changes in the microbiome composition. And there is a reduction in what are known as good bacteria, such as lactobacillus, and an increase in mildly pathogenic bacteria in the gut of these animals. This is the focus of research right now. We're interested in looking at the variants associated with pediatric inflammatory bowel disease in Indian patients, for which we are collaborating with clinicians and doing whole exome sequencing to look for mutations in Indian patients, because according to the clinicians, these variants that are seen in the Caucasian population, where much of the research has been done so far, are not found very prevalently in the Indian cohort. So that is one aspect of the research that we're also interested in at this point of time. I think that should do, Shantanu, in terms of what we do.
Yes, thank you, Sandhya. Dr. Vishal?
There's been some issue with my camera turning on, sorry. Yeah. So I think my work started something, I think around the area when the genomic buzz in oncology was overhyped. And that is when we actually started working on the various aspects of genetics and oncology and started diving deeper into this question. And what interestingly came up, and at that time I remember very distinctly me and Deepak talking, our discussion was that Deepak was talking about aging cells and how they keep them to live longer, and I was talking about a cell that doesn't want to die. And that's where we realized that there are a lot of common signatures of hallmarks of cancer and hallmarks of aging. So we actually bumped across this whole discussion and thought we should take it forward from there. And from there on, I think the whole last, I want to keep it a little focused on cancer specific aging. And what we actually found was during that period, we were actually studying the oral microbiome and looking at cancers in the oral cavity and why are they happening in people with no tobacco. And we could actually find many signature bacteria which actually could connect and show us a kind of a pattern, I would say, to say that there could be a causative role of these people contributing as co-carcinogens in these patients where trauma was a cause of cancer. So that kind of led us to a paper leading to publishing one of the work on oral microbiome and then starting some of the work with gut microbiome and looking at signatures in the area of immune aging and in the area of gut cancers, in the area of understanding is there a signature which we could look at for working and amplifying the immune system. So I think this whole thread led us to many answers, and one of the most recent articles that we've published is on understanding. Now organ specific aging is a very interesting topic today, and for us in cancer that also is very relevant.
Yes, please continue. We can hear you.
Yeah. So I think with organ specific aging, we've now come to a point where we are now looking at and understanding that there are specific markers to predict organ specific aging. And at the same time, we found that through initial studies that we are doing together with IISc and other groups, that there is a connection between senescence and cancer. Now, this is an area which I think needs to be explored and driven deeper. And through this entire conversation and panel, we could maybe discuss more about the connection between senescence and cancer and how the entire metabolic pathway is driven towards cancer in these patients. Thank you.
Thank you so much. That was really fascinating to hear. We'll start with Darshit. If you can give a brief overview of your work, and then we'll dive right into our discussion.
Sure. Thank you, Dr. Shantanu. So, yeah, actually, I think my journey into microbiome and aging comes through two different routes altogether. I had been working on some metagenomics data set on freshwater communities in the Pune city, in and around the city. So we were looking at a lot of, metagenomics related studies out there. And I think that was my previous work. And then after COVID, I kind of got into the longevity science per se. And decided to do something about that in India. That's where kind of 𝗥𝗲𝗴𝘂𝗃 came from. So both the metagenomics and the aging science came together for me. And I decided to, start looking into the human gut microbiome, because when we were working on the freshwater metagenomics as well, we saw that pristine water bodies outside of Pune city had a completely different diversity versus the ones within the city, which was kind of polluted to a certain extent. And the idea was that obviously something similar could be observed in the human gut ecosystem as well. And microbiome was kind of picking up in the longevity science during that time as well. And in 2022, I think it came into as one of the hallmarks of aging as well. That just kind of pushed us towards microbiome and aging together. But as we started working on it, we realized that there's a lot that needs to be solved for India. So we explored different technologies, starting from 16S, short read metagenomics, long read metagenomics. And through the evolution, we have come and understood the data to a certain extent. Now we are trying to strengthen the hypothesis still, multiple hypotheses through our computational approaches. We use a mix of metagenomics, network pharmacology, molecular dynamic simulation, to look at different aspects of how microbiome and human body might be interacting. And soon we'll take it to a clinical stage, thanks to Dr. Vishal. One of the projects is with his team itself on looking at signatures for people responding to cancer immunotherapies and versus those who are not responding to immunotherapies. So that will be first of our clinical adventures as well. I think the path forward will be through that we'll keep looking at the clinical samples. Obviously, throughout this panel, we'll discover so many problems that we are still facing with the Indian sequencing or identifying an Indian microbiome community per se as well. So I'm sure those questions would come up. Thanks.
Thank you, Darshit. So we really have a nice story now. We have a specialist in gut biology. We have someone who has vast experience in oral microbiomes and cancer, and of course, Darshit with having experience in quantifying and characterizing communities with the know-how of the techniques and the technology that's emerging. So the colon, or the hindgut, as it is called in animals, is really one of the organs with the highest densities of bacteria in the body, and a large diversity of abundance, and qualitatively and quantitatively, the microbiome is now emerging as a driver of multiple aspects of health and aging. So can I start with you, Sandhya, to just tell us uh, what is so special about the gut that really, makes it such a wonderful niche for bacteria, and what is known about the function of the gut in the human body and how it changes across age?
So it's very interesting because the gut was the least fashionable organ to study for many, many, many years. Everyone is excited about the brain or the nervous system or even the reproductive system, but gut biology was considered infra-dig and not something terribly exciting to worry about. What is interesting, though, is that whenever the gut doesn't behave properly, we are affected. We're affected in our moods, we're affected in the way that we're physically active. And so obviously the gut is playing an extremely important role in regulating the functions of different organs in the body. Now, if we think about the gut, it's one region of the human anatomy which is not sterile. And that is because the food that we eat is not sterile. The oral cavity is not sterile. And from the oral cavity, the food goes down through the esophagus into the stomach. Lots of digestion happens along the way. From the stomach, it enters the small intestine, which is this extremely long tube that curls around inside our stomach, our belly actually. And then we enter the large intestine, and it's in that region of the gut that the feces is produced, and which is excreted as part of our usually daily activity. Now, one of the important things to remember is a large part of the microbiome research related to the gut has so far been looking at the fecal microbiome. And that's for obvious reasons. The feces is an easy sample to be able to get. So essentially what you do is you collect the fecal samples from individuals, isolate the DNA, and then the DNA is sequenced to identify what organism is in the gut. It's important for us to remember that the organisms present in the gut are not just bacteria. Of course, bacteria outnumber a large fraction of the gut microbiome, but we also have in our gut viruses, we have parasites, we have fungi, and all of these are usually not sequenced when we do the routine DNA sequencing from the fecal microbiome. But there's more and more evidence now that many of these unusual organisms actually contribute significantly to gut function and gut homeostasis. Another important thing to remember is that the environment within the gut changes as we move along the gut. So we have an anaerobic environment in the small intestine, and as it moves through the gut, as we move down the gut, it becomes a little bit more oxygenated towards the colon, the extreme large intestine. As a result, the kinds of bacteria or organisms that thrive in these environments are also different. As I said, what we look at is largely the fecal microbiome, but many of the diseases of the gut originate in the small intestine. And to be able to sample the microbiome from the small intestine is a lot more challenging because that would require biopsy samples. And obviously in healthy individuals, you cannot just request a biopsy sample. You will get biopsy samples only from patients who perhaps have been identified by the clinicians as being affected by Crohn's disease or inflammatory bowel syndrome, etc. So it's important to remember that the microbiome, as we talk of it routinely, is usually the fecal microbiome, but that may not be the most relevant microbiome for the cause of gut disease. Now, the microbiome is, the human gut is populated at the time of birth, while the while the baby passes through the vaginal canal of the mother. Cesarean section infants have very, very few bacteria in their gut. And that is one of the reasons why it's important to look at cesarean babies and make sure that their gut microbiome is populated effectively. For children born normally, they're just through the passage through the vaginal canal, the bacteria populates the gut. And the gut microbiome is significantly changed during infancy up to early adulthood. And that is purely because initially, largely the mother's milk is the diet, and then the diets change as the children grow older. The adult microbiome stays relatively constant, depending on the diet, of course, and that is going to be a challenging factor in the Indian context. The adult microbiome stays more or less constant up to the age of 50 to 60, which is really the period where one considers that one is aging. And it is known that the fecal microbiome of older patients is significantly different from that of younger individuals. Now, when you pass the age of 90, you're considered someone who has shown longevity. And so it's very interesting to look at the microbiome of patients of 100 years or more, or not patients, they're healthy individuals of around 100 years or more. And you will So there is this correlation between a healthy microbiome associated with younger people and a microbiome associated with individuals who've lived beyond the age of 90. But in general, microbiome diversity, or the different types of bugs that live in the gut, decreases with age. And we lose what are called good bacteria. And there is a lot of interest now in trying to see if we can promote healthy aging by providing these good bacteria in the form of probiotics, etc. So I think that summarizes what I'd like to say as far as the gut is concerned and the microbiome in the context of health and aging in the gut.
Right. Just a small point to add, as you were saying, not many of the bacteria can actually be cultivated.
Excellent. So yes. I'm glad you brought that up, Shantanu. Yes. So because these bacteria or other organisms, for example, are what are called non-cultivable, the only way that we get to know what the bacterium is by sequencing the DNA, which is what is done during microbiome analysis. What the state of the art now is that knowing you have a particular bug in the gut, you have available whole genome sequences of many relatives of this organism. You can look at the whole genome try to identify genes which are important for example, producing specific metabolites or producing specific compounds which are beneficial. You can now try to reconstruct this in an artificial organism and try and feed it back. Now, most of these studies so far, of course, have been done in animals. These are animal studies. But I think this is the dream or the goal of human microbiome research. It's quite possible that at some point of time, we might even be able to develop culture conditions that mimic the gut environment a lot more. And therefore, we might indeed be able to culture some of these organisms and start looking at them when they're alive, rather than just going by predicted sequences that the genome has.
Right. So this is a nice, actually, a segue into, since we're talking about aging, let's start from the very beginning. So Dr. Vishal, with the increasing changes in lifestyles, and as Sandhya was suggesting, more women opting for cesarean deliveries, also decrease in the prevalence of breastfeeding and reliance on more synthetic diets as babies and infants. How do you think that is now affecting the oral microbiome? And do you see subsequent consequences of that in pediatric cases, as well as on the general process of aging?
So, Shantanu, here, most of the data that we've extrapolated is from the West. And that is where I have been a little concerned that all our data need not be a reflection of the West data, because the gut is something so specific that 100 kilometers away from Karnataka, from Bengaluru, you will have another gut microbiome signature. For every 100 kilometers, maybe the signatures may change. It may be actually very interesting for us to actually look at this entire thing from an Indian perspective and looking at it from, and that's why when we did the first study that we had, we realized that these were original signatures that had not mimicked the West. So I see a great opportunity here for us in terms of going deeper down and understanding the patterns of expressions, because the pattern of healthcare here is different compared to the West, where most of the delivery is vaginal delivery, a normal delivery, versus a cesarean section, which is done routinely as a part of the culture here. So it's going to be very interesting to see and correlate this data at a much larger, I would say at a much zoomed out level. So I would just like to start by saying, when we started talking on longevity and I was looking at global data, they said centenarians, there's this big buzz about centenarians. And I looked at what's the global data on centenarians. We had about 90,000 people in USA, 70,000 in Japan. And I was curious to know what's India's number. We naturally don't have a proper structured data. So I went back to the government and I requested, saying, I want a little more data on this, which they had structured district-wise. So they gave me a data which was quite surprising for me. Karnataka. I took the Karnataka data and I found we have 22,467 centenarians. Now, think of it. I mean, what are we talking about? If we have this data, of course, we need to validate this, we need to prove. But I think the initial dipstick of a home-to-home survey data from the government showed this is the number of centenarians in one state, which means technically, if you have to put these data together, we will have more answers to give to the world on a blue zone or any other thing than compared to any other company or a particular group that is actually trying to answer the question. I think we now need to seriously take a peep into our own guts, understand what's happening there, what are the signatures, and how do we actually use this data for science, for commercialization, for creating new solutions, which could be not only for the elite, but also spectrum across age groups, making it more inclusive. Longevity should be a more inclusive subject.
Right. Centenarians seem to be much celebrated in the West. In India, we hardly hear of them, right? And hardly bother to, sample them or get information from them as to what their secrets are. So it's really a fascinating number. So Darshit, you've been analyzing many people especially in the Indian context, right? So what do you say are key signatures of microbiome changes associated with aging? And what are the major correlates that you find from the young to the extreme centenarians?
When we started sequencing so many folks throughout India, we really thought we would come to that answer very soon, but unfortunately not, because I think the the diversity of the population itself throughout India. It's just immense. It is very difficult to put two people in the same bucket, like I think Dr. Vishal also just mentioned. Additionally, what we have also seen is that one thing that definitely came across throughout our cohorts was that India is definitely a 𝗏𝗏𝗃𝗃 dominated gut. As we see, most of the Indian people that we have, they have a very high abundance of 𝗏𝗏𝗃𝗃 in their gut. How has that come about? It's really difficult to answer as of right now. It might be genetics, because we have known that genetics do play a role in which microbial species kind of get a chance to harbor in the gut itself. But I think that is definitely one big factor that has come up. Other things that we usually see is that people who are native Indians, which is like through generations they have been living in India, they have a very high abundance of enterotype 1, which is more of a fiber favorite kind of a genetic setup for microbiome. But I think folks who have shifted out to India, let's say expats from other countries, people who have married into India but are actually foreigners, we actually see that they are more bacteroides favorite. So it is really interesting, that data point, that genetics do support what kind of species that will be hosted in your microbiome, no matter what dietary habits you are following on an everyday basis as well. So I think these two very interesting signatures have come up for sure in the data set that we have been looking for. But I think just coming back to the centenarians aspect that Dr. Vishal and Dr. Sandhya, both of them mentioned, is also that I think recently one of a very excellent opinion article was written by someone from the scientific longevity community of the world. And they mentioned that deciding if a person is centenarian is an even bigger a challenge. In our early days, we tried to do a centenarian project per se, where we reached out to 20 odd centenarians in rural areas of India. And first of all, the stigma to collect stool or get it collected from them in itself is a bigger challenge than any other technology or any other clinical correlation or statistics that we might talk about. So I think that stigma was much of a hindrance. Additionally, when I tried to give this cohort to the International Centenarian Consortium, which is actually a cross-continental team of folks working on centenarians, it includes group from Sardinia, Japan, Iowa State University in USA, and all of them had just one requirement. You need a solid paper proof for proving that a person is centenarian, which is another challenge that we might find in India, which we don't have, thanks to colonialism in a very sarcastic way in that case. But yeah, I mean, these are some of the challenges that we have seen. Also, I think the definition of healthy microbiome itself is a bit shaken. So it is very, very, very difficult to categorize a person as a healthy person. Do I consider non-chronic disease as a healthy person, but they are having bloating every day? Do we consider that healthy? So these are some of the issues we are still battling out, so that I might not have a clear answer on how is the microbiome progressing with age. But I think we might never come to that answer, is what I'm expecting. I think there'll be buckets of age group, and then we'll have to start looking at that.
Right. So I must say that questions have been pouring in. We have a huge list of questions, and I will try and fit in as many questions as possible. But before we also start, there seems to be a lot of questions on what is in a healthy gut microbiome? How can we change the microbiome to make it more healthy? Um, uh, so also, Dr. Vishal, do you think healthy aging is a misnomer, or is there something called healthy aging? And what is the definition and how does the microbiome fit into it?
So starting from the age-old definition of WHO, which defines health, which is physical, mental, spiritual, blah, 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 all kind of a thing, to the today modern era of Gen Z, Gen Alpha health kind of a thing. I think the definition is zoom past changing. That's what I'm understanding. And I think it's very interesting to look at now the term health span. They're not talking about lifespan and centenary. They're also talking about health span and quality of life. And I think from that perspective, health is a moving target. Now, I think we've been delving deeper on this to say that health now, as we go deeper into these subjects, we realize that health could be organ specific. So a person could be overall healthy, but his liver could be actually not that healthy. So now I think we have to move towards a system-specific aging and health versus an organ-specific aging or health. That could give us more meaningful insights into focusing on the larger picture of longevity. Because on a macro level, I don't think we have one single answer today. And mental health being more elusive than all of these in the area, and yet gut microbiome being at the center of mental health. So it's so complex, while so intriguing and interesting. And at this time, I think in the mantra of all of this, the one thing that we surely know is what doesn't work. What works, I'm not sure we know. What doesn't work is definitely what we know is what is all available in Amazon and across the counter doesn't work. I think in a very cautious way, I would like to say this, that at least that doesn't work. Now we've been discussing this with IISc, with Darshit, with all experts across, to say that the one clear trick in this is, I think, like take a probiotic and it flows through and through your gut. And I think it's like a complete straight pipeline from entry to exit. So what is it that gives the stickiness factor is something that we've been questioning and trying to figure out and write something on it. And that's absolutely phenomenal to look at, because it's the lipid layer there that is actually making the trick of stickiness. So there's a lot for us to answer today on what works, what doesn't work, how are we going to make it work. I've just got my official approval for doing fecal microbial transplant in HCG for cancer patients who are not responding to immunotherapy. So I'm sure in the next coming days, I'll be able to hopefully give some more insights into FMT, and is that the answer that's going to come up apart from just customizing probiotics. So look forward to some of the next coming days of advancements from India.
Yeah.
Right, as you said, if only it was so easy to go to Amazon and buy something and reverse agent. If only we all wouldn't be here. But there seems to be a lot of questions on cancer and microbiome. And starting off what you were also saying about the role of the oral microbiome and trauma in causing oral cancers, as well as colorectal cancers, and how our diet is now making individuals more susceptible to cancers. So some of the questions are related to this connection, and also how much does diet, or in terms of oral hygiene, are those important factors, the food that we eat, the kind of products and the insults that we subject are, of course, tobacco and cigarette are well proven, but there are so many things, which are within our realm of hygiene and care. How does that affect overall the connection between aging, microbiome, and cancer?
So to put it in a short nutshell, I would like to say that as we were studying this, we realized that senescence itself was a very interesting subject. Now, organ-specific senescence is something that we are studying more closely to understand that there is enough of intriguing and I would say data that is pointing out to say that it is these senescent cells that actually are actually I would say secreting a lot of cytokines which are pro-inflammatory and influencing the cancer possible stem cell, I could call it. I don't have the perfect word for it, but what I'm saying is that there is definitely a metabolic pathway in this entire connect between senescence and cancer, because there seems to be an affinity metabolically for the senescent cell to move towards a cancer pathway using the aerobic glycolysis pathway. And there are very, very interesting papers on this, which people should deep dive into to study and see how a senescent cell's metabolic pathway shifts towards a lactate pathway and aerobic glycolysis, which could be a precursor for cancer. That's one aspect of connecting aging, senescence, and longevity. Now, the other part of it is oral microbiome is a part of an extended story, because we have skin, we have vagina, we have oral cavity, and we have the gut. Now, all of them in some way have specific signature, and the same thing is for the oral cavity. And in the oral cavity, what I've been digging into is there an oncobiome there? Is there a set of bugs that are actually contributing to being an oncobiome and not just a probiome? And that's something that we are actually getting some very relevant data to point towards, because if you are able to crack that, then we actually have some meaningful solutions for oral health, which talks about not using antiseptics or these Listerine kind of mouthwashes, Listerine and other kind of antiseptic mouthwashes, versus using probiotic mouthwashes and using probiotic health for the oral cavity. And that could be a major game changer that we could actually be showing the world in the coming years. So I think the list of discussions are long, but I think the hallmarks are similar, and the connect somewhere for us in the initial data seems to be a metabolic shift in this entire thing from senescence towards cancer. And that could actually lead us the way towards, again, coming back to organ-specific aging, organ-specific senescence, and organ-specific cancer predictions. If we are able to pick this up and actually able to correlate the genomics with the contagious genomics, that is our microbiome, I think we will have much relevant data. And we started at HCG collecting this data in a simple box. And I'll just close with that. We have a patient's tumor histopathology, which depicts the tumor's nature. Then we have the patient's full genomics, a whole exome or an actionable panel. This is the tumor signature. We also started collecting the blood immune profile, which has all the cytokines in the patient, and we've created an immune score and correlated that with the gut microbiome. Now, all of these four put together, there is 2 parts of it which are related to the tumor, 2 parts of it related to the patient. Using this, we are trying to put a structured plan to understand and give the patient to say, with your immune score like this and with your gut microbiome like this, what should your diet be? And how could we customize your diet towards activating your immune system based on this particular thing? And we have some good longitudinal data to show that this could be actually changing. And that's exciting for us. So we will need to look at this from one single nutshell and not isolated silos of just looking at a gut microbiome or something like that.
Mm-hmm.
I think I'll stop here. Thank you.
Yeah, is it safe to, just to summarize for the audience, to say that the cause between colorectal cancers or many cancers and microbiomes are still in the early infant stages? We have some correlations, but the cause effect is still in the early stages, and a lot more needs to be done to find which bacteria, which metabolites, which secretory factors are all causing this. Will that be right to say?
Yeah, that would be for the oncobiome. And I think that's something far-fetched right now. But before that, I think for working on the immune system, there is no debate. I think we should use the gut microbiome to structure, channelize, and plan curated diets. And that's where the next problem will be for all experts, including Darshit, to kind of give an AI planned structured diet based on Indian palate for every 100 kilometers using your gut profile, you know?
So the $1,000,000 question, what should we eat?
My answer has been that in the next 10 years, people will die more of eating than of starving. I think the lesser you eat, the better the gut microbes are going to survive.
Yeah.
Sandhya, there are some questions based on the gut health and the microbiota and age-related diseases. There's also a question related to women's health saying, can menopause cause leaky gut? And how does gut bacteria affect other diseases such as obesity, diabetes, heart disease? And is there any role of post-epigenetics in regulating gut and microbiomes?
You know, what we've discussed so far is the bane of all researchers, and that is correlation or causation. Now, what we've looked at and what we've been able to do so far, I posit, with respect to the microbiome, is a lot of correlation. For example, let's take diseases of the aging, which you wanted me to speak about: Alzheimer's, osteoarthritis, menopause. We know that the microbiota is changed in Alzheimer's patients. We know that it is changed in patients with osteoarthritis. We know that it is changed in menopausal women. The question is the change causing the effect in these patients? Or is it because these patients have succumbed or been subject to these issues that the microbiome is changing? And that is going to be the most difficult thing with respect to microbiome research. And we will get wonderful correlative studies, I'm certain of that, along the lines that Vishal mentioned. But whether Darshit will ever be able to translate it, even using AI, to come up with something that is going to work is the challenge. Now, the reason is this, and this I think Darshit alluded to. all bugs don't colonize the gut, even if you give them orally. We know that it is the environment of the gut that is conducive for certain bugs to colonize. And if you have a patient who, for other reasons, has an altered gut environment, however much microbiome you might give them or probiotic you might give them, Lactobacillus is never going to colonize. And we've seen that in our animal models as well. So this is something that, you're either going to have to tweak the host to allow the bug to colonize, or you're going to have to give up on that because the bug cannot colonize given the environment of the gut. I was reading an interesting article today where the last sentence is something that I'd like to quote because I found it very relevant to the discussion as we have it right now. We know that the microbial composition changes as we age. We also can correlate a changed microbiome with the health of the particular patient. Can we use then the microbiome as a way of geriatric care? So if you see that the microbiome composition is changing in an older individual, is that a biomarker for something that may go wrong later, which perhaps could be checked at its inception? So I found that very interesting, that a microbiome change is a biomarker for something else. It's not necessarily the cause of what it is. It's not that, a bad microbiome is causing an older person to get constipation or diarrheal disease or, poor muscle tone. But when you see that correlation, are you now going to be able to say, okay, that is associated with this particular disorder? Can we try to prevent that using therapeutics or drugs or changing diet or whatever? So I thought that is maybe where we're likely to get. There's a lot of excitement right now about the microbiome and what it does. And, research is really very exciting when you read some of the really wonderful papers that are coming out where you can identify a particular pathway in a bacterium that is making dopamine, which is now going to the brain and alleviating, some issue or other. All excellent research, but whether it's ever going to be able to be therapeutically, adapted is something that we're going to have to wait and see. And I think I'd also want to say one thing, that, aging is inevitable for every one of us, right? I mean, we have 4 different people in this group who have aged to different extents, and it's going to be something that's going to happen to all of us. The question is healthy aging. What are the kind of things that we can do to stay healthy? Is the microbiome going to be able to tell us that something may be going wrong, and therefore we need to correct some behavior or our diet or something or other, rather than it actually being the cause of what is going to happen to us later?
Right. Sandhya, could you briefly expand on the role of viruses as constituent members of the microbiome and what they do and don't do?
Yeah, so the viruses in the gut may not even be free-living viruses. They may be actually bacteriophages that live within the bug that's present in the gut. When we sequence the DNA, we would just get a DNA sequence. We wouldn't know whether they were free-living viruses or viruses that live in the bug. Now, we all know how important viruses are in bacteria in which they live. I mean, the whole CRISPR-Cas system that we now know as being a way of generating mutations, etc., all came out of an understanding of the viruses that infect bacteria. There are likely to be viruses that live in the gut independently, but viruses by definition can't really survive on their own. They need to be within the host cell. So they may be colonizing the host epithelial cell, which gets sloughed off in the gut. I think one thing I'd like to mention to the general audience here, the gut is the most replicating tissue in the entire body. We renew the entire gut lining once every 4 days. So it tells you about the proliferative capacity of the gut to be able to regenerate itself. And as it does so, the cells at the top of these villi that are there in the intestine slough off. Many of those sloughed off cells may contain viral genomes, which have now got integrated into the host DNA. So it's going to be, I don't think we're as advanced in understanding the viriome, That's what's meant by the viral microbiome, the viriome. I don't think we're as advanced in understanding the role of the viriome in aging or in gut physiology. As I said, we have fungi that live in the gut. That's another black box.
Right.
We have certain parasites that live in the gut. So I think we may be focusing for technical reasons only on the bacterial component, but it's quite likely that many of these other things will have secrets which need to be seen.
Okay. Yeah. So Darshit, there are some questions on how can we monitor our gut microbiome on a routine testing basis? How reliable are they? How often should they be done? And is it really useful? So, the recommendations or the interventions that one would get from a microbiome report, not without visiting a doctor or without getting a medical opinion, just getting an over-the-counter microbiome test. What can you say about that?
No, I think first of all, microbiome is way too generalized as a word now, especially in the preventive care, prognosis, or diagnosis industries, right? Because if we actually see, there are multiple ways to look at microbiome, and each technology has a certain kind of an upper hand and a lower hand on a certain thing. For example, if someone would come to me for a preventive screening of microbiome, but is really looking to identify something like a H. pylori in their gut, right? Metagenomics might not be the best way to do that. I think there are pathogen assays developed for that. So I think every kind of problem or prognosis that we want to make, the kind of test or the testing methodology used is completely different. I mean, if you are a healthy individual, you are on your preventive journey and you want to optimize yourself, then yes, a non-biased metagenomics test, like something what we do, is perfectly feasible because that gives you an overall picture of your microbiome. And based on that, a certain dietary recommendations that we can give. And those dietary recommendations are mainly based on the logic of which bacteria would survive on what kind of substrates. And I think one of the interesting questions that you shared as well was that can we kill certain microbes with the food? I think, yes, there are some broad spectrum spice families which can help in killing certain bacteria. But I think a better strategy towards that would be starving off some of these bacteria which are actually growing. I think FODMAP diet comes from a completely that kind of an ideology. That if bacteria can grow on fructooligosaccharides, just cut them out, right? So it's something like that. I think it's the starving plus killing while allowing the growth of some other bacteria. I think these are the three strategies that are given in the microbiome-based diet recommendations. And it's a little bit of a problem in terms of following, because adherence in India anyways is a big problem no matter what health solution that you give anyone, especially with microbiome diets. It's quite complex. I mean, we feel it ourselves because me and my wife, we cook dinner at the same place, but she is used to millets, but my gut is not able to get used to millets. So it gets complicated for sure. And I think an easier way has to come out of this microbiome based personalized diets. But I think if followed to the T, it will give some relief to the patient in terms of the common symptoms that they are facing. But I would still say that if you're going for diagnosis per se, that do I have a certain pathogen or not? I think techniques like RT-PCR work better. But if you're going for preventive health optimization, healthy aging journeys, techniques like metagenomics would give you a fair understanding of how your microbiome is, right? And I think covering both Dr. Sandhya's and Dr. Vishal's notes is that a strong hypothesis that I have conviction in is that if you're trying to diagnose or prognose something, maybe go a little bit reductionist, try to find out that 1 or 2 or 5 species that might be contributing to that condition. But when you're actually going for interventions, it is not simple as A to A. If this bacteria is missing, I'll just give a probiotic and it will work. That's not going to work. I think it has to be a more ecology approach when it comes to intervention, but a more reductionist approach when it comes to diagnosis. And I think the choice will be very much different for every patient in terms of what test to go for. Am I looking for something and what is the solution that I'm seeking?
Right. Dr. Vishal, is there any telltale symptom of dysbiosis? And especially in Indian populations, as you age, the population is more susceptible to something like vitamin deficiency, right? Many of them being attributed to dysbiosis or changes in the gut microbiome. So over aging in the Indian context, what should individuals do if they suspect they have dysbiosis? Are there some remedies that you suggest? When should a person see a doctor, especially an aged person, where they think there's something is not right in terms of their gut health?
So I think, Shantanu, if you look at the current status in the OPD that I sit in, every single alternate patient or every patient has some dysbiosis because they're going through chemo, surgery, radiotherapy, the nutrition is shifted. So there is something. So I almost every day on a day-to-day basis see dysbiosis. In a generic level, I found two words very fancy today: deficiency and allergy. These two are Google doctors' main contribution to the generic society. I see everybody coming up with this and throwing up these two words. Sir, deficiency hai aur ya pe mujhe allergy hai. Now, the best benchmarks we have seen for this today are some blood tests which are using IgE, where they give you some 300 allergens and tell you you are allergic to everything that you are under the sun, which again too non-specific, versus the gut microbiome, which is trying to actually correlate this with the allergen. So I still don't think we've cracked the full puzzle here of dysbiosis. But one thing we know is COVID vaccine or virus has definitely contributed to dysbiosis. Very generically, today, if you look at the pattern of global data, if you have been infected with the virus or taken the vaccine, dysbiosis has set in. There are a sudden shoot in IBS, there is a sudden shoot in gluten intolerance, sudden shoot in lactose intolerance, casein could be another cause. There is suddenly a steep rise in dysbiosis. Now, we definitely know post-COVID, there has been something that has happened to the microbiome. It's a contagious genome, and so naturally something has got affected there. We need to, I think at this juncture, put some little point, I mean, maybe this kind of a consorted group could put some of our energy to understand what actually shifted. And I don't know if people like Darshit's team has the data of pre and post. It's worthwhile studying what actually shifted. Because obviously there are people way far more coming up with IBS today and Crohn's disease and allergies and lactose intolerance and gluten compared to what we saw before COVID. So obviously there is something happening. We need to get the correlates of the blood IgE allergens versus the microbiomic signatures of allergens and deficiencies to correlate this. And like Sandhya said, I think correlation is not causation. See, like you do 100 people, all of the 99 will be vitamin D deficient and they'll be starting to pop up this pill. Is there a correlation with microbiome? Nothing. So, I mean, I think somewhere we are overdoing technology. Like I often say that artificial intelligence is not a replacement for natural stupidity. So, you know, you just can't go on doing tests and finding some solutions and then start treating yourself, which people like today, and they kind of feel very assured that, okay, there's something that you're doing out of it. So I guess there are more questions than answered. I think the next session, maybe we'll come more prepared with answers, I feel.
Right.
A decade
So
down
there
the
are
line.
Yes,
there are
Sanjay,
a
go
few
ahead.
questions posted in the chat box as well.
Right. I'm just coming to those. I have one in front of me. There's a question again on what is the role of gut microbiome in reducing the side effects of anti-cancer drugs, and also um, having a particular gut community, does that make you more resilient in your coping with chemotherapy or radiation?
So to the first answer, see, very importantly, forget the gut microbiome. Food is important during chemotherapy. What people most of the times forget to understand is food itself as a simple formula is often neglected part of the story. We are all busy killing the cancer cells, which comprise of maybe 5% or 10% of your body. And you've forgotten the 90%. And you're saying, you know, a lot of the times the consultation is like a short 2-minute consultation where you say, kya kha sakte hain, kuch bhi kha lo. Now, I think that is where onconutrition has come into place. Like I have a structured onconutritional program. I do not tell them these words, kuch bhi kha lo. I feel it's important to spend time and telling them. Now, what is this kuch bhi kha lo? Say, what is the structured oncology diet during chemo or any cancer therapy is high fiber, moderate fat, less carbohydrates and less sugars, more amount of water is a generic mantra that will give rise to a good microbiome. which will actually help in rejuvenating your old microbiome and keep you healthy. And that's good enough to combat the side effects of chemotherapy. In a very general way, I'm saying. Now we are moving towards FMT and other things as a part of the clinical studies, and we will have subsequent answers. But till now, I think the answer is more structured on giving an oral giving a proper structured dietitian plan, which is high fiber, and protein rich and less amount of carbohydrates and more fats. That would be more of a generic answer for this microbiome rejuvenation therapy today.
Is there any connection between gut microbiome and autoimmune disorders?
I think all of us would agree that there is. I think there is no debate about this.
I think expanding that answer will be difficult, but I'm sure there is.
There will be.
Right.
Yeah.
There will be.
So,
and Crohn's disease itself is an example for this. That we've been able to. We've never thought about IBS and Crohn's disease cure until FMT came into the picture.
Right.
There's one more question. More recently, Mayo recently published that they are able to predict chronic conditions occurrence 5 years down the line based on microbiota profile with a 70% accuracy. What can we learn from it?
This is what I said, that the microbiome could serve as a biomarker for something that's likely to happen down the line. And, rather than it actually causing what's going to happen down the line, there are certain changes that you might see which could result in a particular, problem later on. So I think that's what we're likely to get out of all of this. That if there is a microbiome change, it could be correlated with a possible outcome, because host genetics also has an important role to play. And it's quite likely that even if you have a lousy microbiome in your oral cavity, you don't get oral cancer. There's a chance. 80% might, but you still have that 20% that may not. So we're only going to get correlates and not absolute proof. Sorry, Vishal, you were saying.
No. I just wanted to add to what Sandhya said, that I think in the current context of the microbiome and looking at this, there are a lot more, like take tobacco for example. Tobacco causes cancer, but in certain of the cancers, it predisposes you to cancer. And I think that's the point that we need to make a distinction here, that not all of that data makes you cause it, but some of them may predispose you to a certain condition. We are currently writing a particular paper called the circulating microenvironment. And I think the microbiome and the contagious microbiome could be a part of this extended circulating microenvironment. And Darshit's team has actually got us some initial data to say that there is a blood microbiome that actually is giving us some crosstalk. How relevant it is, we are far from it, but I'm definitely sure there is a tumor microbiome, there is a gut microbiome, and there is also a blood microbiome. So what are they going to do? I think a long story.
I think if I may just add to that, I think both on the disease question and what Dr. Vishal just mentioned, I think even the Mayo Clinic study, as soon as they'll expand the population from the one that they have tested, the hypothesis will kind of start falling down. You'll see that so many companies throughout the world are trying to give you a disease prediction score based on microbiome, which personally I have not been very fancy of in our own test, is because it's very difficult to do that. And I mean, 70% accuracy, they're themselves disproving that the prediction is not so strong. But in any case, I think this disease prediction is often happening based on correlation, association, positive and negative associations and correlations. And I think this is not going to stand the test of statistics when it goes to a continental level of pan-continent data set per se. And I mean, interestingly, what Dr. Vishal also said, we tried our hands with blood and tumor tissue microbiome. But I think everyone needs to be a little cautious after what has happened with the company called Microbiome, which spun out of Dr. Rob Knight's lab. And we have seen that here is the real challenge of technology. Forget ML, forget AI. The bioinformatics itself is not so advanced as to solve the reasons for microbiome data. They have seen that there was a lot of noise in what they had reported in terms of the tumor microbiome or a blood microbiome, and they had to retract 32 odd papers from Nature. So that's a big setback for the tumor and blood microbiome characterizing communities. And I think we need to be more conscious on the reference databases, the sequences for bacteria which are there.
I think these are the challenges with analysis, which, as the field evolves, we realize more and more things. We've recently been studying studies demonstrating what is called as a ketome, which is the microbiome of the kits that we use for DNA extraction and things like that. So there's all sorts of contamination everywhere. And, it's sometimes it's inevitable that we cannot escape microbial DNA from our samples and sources. So there's also been a lot of talk recently, where they sequenced the microbiomes of tribal communities, especially in the West. And they showed that they found that this tribal or the Amazonian rainforest communities, gut microbiomes are very different from what is the Western microbiome. And they've blamed it all on the diet and the lack of exercise and the lifestyle. So over time, we see the microbiome being blamed for everything, giving credit for everything. So I would like to get your thoughts, the three of you all, on what you all think of this ideal microbiome. So is now it's being come to believe that the Amazonian rainforests have the most ideal microbiome, and it's for that environment. It's not they haven't faced pathogens and parasites for hundreds and thousands of years. So a microbiome will only make you fit for a particular environment. But this craze of idealizing a microbiome, let's aim for this because the rural folks have this or the tribal folks have this. What do you all think about this?
So it's like asking, what is an ideal human being? Is it Donald Trump or Kamala Harris? So,
we'll find out soon.
Yeah, so exactly. So I think we have to think about host genetics here, which is extremely important in shaping the microbiome. It's well known through studies in the West that the Mediterranean diet, for example, is supposed to result in a wonderful microbiome. People can live forever on the Mediterranean diet and things like that. But, Indians are doing quite well. We're the most populated country in the world right now. So in which case, presumably, we have a way of surviving as a human race, as Homo sapiens. Whether we're the ideal human being at all, and we harbor the ideal microbiome, I think that's a point for debate.
Right. I think if I may just add to that, I really don't think so there'll be an ideal definition. I mean, coming back to the first question in the discussion happened with that. I think there are politically divided states and countries, then there'll be microbiome divided states and countries. There'll be zones where people of certain age group, of certain ethnicity, of certain environment, be it air quality index that is there out there in that. And I think those little zones or pockets will have their own ideal microbiome communities if we have to go to that level in any case. But honestly, I mean, if any one of us urban dwellers would take a FMT from, let's say, a Hazda community in Africa or someone from an Amazonian rainforest, it's going to prove disastrous for us, I think. And we really have to look at the species structure as well. I mean, the species that are identified there versus the species that are identified here have very different genome assemblies as well. So we are completely neglecting that factor. Oh, we got a bifidobacterium in US and in India. Great. But the bifidobacterium are different. What about that?
Yeah, absolutely. So we are almost, it's 5 o'clock, so it's time to wind up. But I would like to end with a small discussion because we have a large student audience just now. So a lot of us don't actually get a chance to study the human microbiome. But Sandhya, you've been studying the microbiomes of mice for many years now. And what do you think are the takeaways, the pitfalls that one should be careful when you extrapolate a mouse microbiome study to humans?
Yeah.
Or what are the lessons learned from mice?
One of the major differences is that the mouse gut itself is very different to the human gut. We've looked at the mouse receptor that I'm interested in versus the human receptor, and we find fundamental differences in biochemical activity, which can all be correlated with the mouse diet and the human diet. Our diet is far more rich in water and fluid. Mouse diets, if you think about mice that live in the wild, they hardly have any water there. It's all dry food or dry nuts or whatever it is that they eat. So the gut has adapted to that intake of food and environment. Consequently, the microbes that live in the gut of those animals is also different to the kinds of microbes that live in the human gut. So again, we can, for example, do something to these animals and look for a change in the microbiome. And we can say that with that intervention, the microbiome altered. And therefore, in humans, if you do a similar kind of intervention, you might see an alteration in the microbiome. But a direct correlation between the bug that's there in the gut of the mouse versus the bug that's there in the gut of the human, I think is stretching stuff, exactly as 𝗔𝗴𝗶 mentioned. If you were to look at the whole genome sequence of Bacteroidetes sitting in the mouse gut versus in the human gut, there would be a number of changes that you would see. And but the species or the genera remain the same. So I think we need to be cautious there.
Yeah, all right. So Darshit, given the panels that you have for microbiome analysis and biomarkers, there are some questions on what is the general procedure of stool to result. So can you walk us through the pipeline of how you do it?
Yeah, I mean, it's pretty simple to be honest, except for the fact that you have to look at your own stool and collect it. Rest everything seems quite very simple beyond that. So it's pretty much a kit that is to be used at home. And I think again, coming back to what you also discussed, Dr. Shantanu, we have to have microbiome analysis for this kits done to discount that from the final patient microbiome as well. But yeah, I mean, it's a kit at home. There's a DNA stabilizer tube, and you kind of put your sample in it and we bring it back to our lab. We do the NGS analysis followed by bioinformatics analysis. We look at the functional parameters, we look at the taxonomy parameters, and then we kind of generate the report out of it. It's a very simple kind of a walkthrough that we have for the microbiome test today. One of the challenges that we have faced in that is that in summer times, if you are collecting samples from, let's say, a Delhi or a Rajasthan, where temperatures are going 45 degrees and above, those DNA stabilizers are kind of not working to a certain extent. This is a problem we realized only when we came down to India and we started using the same DNA stabilizer as the foreign countries used. So I mean, challenge really starts at this point, at sample collection. Forget about all that, which is why I always say whenever I'm in a microbiome, discussing microbiome, I'm like, keep MLAI out of this conversation at least for 5 years. We still have a lot of fundamental things to do. And I think another challenge is that across different microbiome companies, there is not a certain standardization of the technology itself. I keep talking about this. Everyone in the genomics industry started talking about depth and coverage, at least after 5 to 7 years after the boom of genomics. And I think this is a conversation we should have since day one in microbiome, because there are companies who are sequencing less than 500,000 reads and they're giving results. At the same time, there are companies who are generating billions of reads and then giving a result. So there'll be a stark difference between what you find in them. So I think the standardization in the sequencing depth, the coverage, how you're analyzing the results, how you're polishing the results, I think everything makes a difference. And I think the industry needs to come across with certain guidelines for that. Or if not the industry, I mean someone from the policy making side of things. Otherwise, the trust will be lost in the overall microbiome diagnostics.
Right, right. So there's a lot of companies doing a lot of things, giving a lot of results. There's a huge black box there, right? So this needs to be one needs to be careful about it. So final words, Dr. Vishal. There are a lot of students in the audience. What advice would you give them as individuals, as people who want to take care of their microbiome, as students who study oncology and microbiology, as students and as individuals, as citizens? What advice do you have?
I'll start with the good note, saying that the worst vice that a person can have is advice. So I think my advice will be very limited to you. But overall, I think it's a good, I think it was a good interaction. I think there is a lot of messages for all of us in this. I think the part of one is eating less rather than eating more is a big mantra today. And I think that's a very successful mantra. So the idea of health span will begin from minimizing the eating and looking at more of nutrition than of diet. So I think we'll need to balance these two words, which cannot be used interchangeably. And I think for the student population also, I think today we are, there is an instant rush, there is an instant era, there is a more era, and there is an unlimited era. These three are the hallmarks of the next, this Gen Z, Gen Alpha times. Now I see that you are looking for instant food, instant pizza, instant coffee, instant marriage, instant love, instant blah, blah. And, one buy one, get three free, and you're looking at unlimited food, which I think is the worry that we are having. And I think that is where the Gen X wisdom may help a little bit, and whatever the other generations, because that's the one that's going to actually connect the gut to the brain, finally, to make sense. AI will not solve this whole problem. It's so as I said.
Right. So for those in the audience who have interpreted you as saying, eat less, I should be fasting more.
Yeah.
What is your comments on the young and the old fasting intermittently, brazenly, without thought for
I mean, I think
care
these
of
are all
health?
page 3 words that have been used, this intermittent versus this. I don't know much
Right.
of those, but I'm just saying that minimalistic approaches are the way forward. That means that based on your BMI, based on your overall thing, what's your current caloric requirement, and having a nutritionalist, investing into a good nutritional program and reducing your health insurance would be the more wiser approach than reaching the pinnacle of NCDs with the peak of all the premiums that you've achieved. That would not be something, I don't want to sound too preachy on it, but I always look at it this way, and I tell people, look, you're actually spending so much to meet me here. If I give you a simpler solution, you find it too simple. If I tell you breathe, if I tell you drink water, if I tell you to eat healthy, you're saying, oh, that's too easy. Tell me something more complex. What can I buy in Amazon or somewhere else? So we are moving towards I would say, an extreme consumerism approach. So I think post-COVID, all the more relevant for the youngsters to look at, I think from a research perspective separately, but from their own personal diet perspective, you will be controlling your health. And I think that particular part is going to be the most relevant, most valuable thing that we can hold as a lesson from COVID. So that responsibility lies beginning with food, water, air, and activity. These 4 are going to be most relevant for us to survive the next few decades.
On that very encouraging note, I think we'll end here. Lots of fine discussion today. I'm sure the audience has also gotten answers to most of their questions. I've tried to fit in as many as possible. But really, thank you all. Thank you, Sandhya. Thank you, Dr. Vishal, Darshit.
Thank
Thanks a lot for
Pleasure.
taking time to be here, answering the questions. Thanks a lot. And for others, take care and bye-bye.
Thank
Thank
you.
you.
Thank you, Dr.
Thank you.
Shantanu. Pleasure.
Bye.
Thank you, Sandhya. Thank you, Shantanu.
Thank you,
Thank you.
Vishal.
Bye.
See
Thank
you,
you.
Darshit. Bye-bye.
Yes.